Okay, we shall continue. Okay, so yes, so this is where we were, the acrosome reaction and the fertilization stages. So as this sperm goes up to the fallopian tubes where it's best, where the conditions are optimum for fertilization to take place, once the sperm now meets with this oocyte or this egg, you'll find that there is what is known as the acrosome reaction that has to happen. So the acrosome reaction is induced by what are known as zona proteins ZP1, ZP2, and ZP3 after the sperm binds to the zona pellucida. Okay, so once the sperm meets the egg and then it uh, binds to the zona pellucida, there are zona proteins, okay, known as ZP1, ZP2, and ZP3 that induce this acrosome reaction. So what is this acrosome reaction that we are talking about? It is simply the release of the acrosomal enzymes. Okay, so those enzymes that add the acrosome there, where we have removed the glycoprotein coat, will now be released. Okay, and then the enzymes are basically known as the major enzyme there is known as acrosin. Okay, so these acrosin enzymes are rather more like enzymes that help degrade this oocyte membrane, the zona pellucida, so that the sperm can penetrate easily through those three layers, and then fertilization can now occur. The fusion of the gametes, the fusion of the nuclei themselves. So that's what we are talking about when we say the acrosome reaction. It is induced by zona proteins, these zona proteins, ZP1 all the way through ZP3. And once it occurs, the acrosin is released, and then fertilization can now start. And then this fertilization is in three stages. Okay, once the sperm penetrates, you're going to have these three stages occurring. Number one, penetration of the corona radiator. Okay, so this is so let me just make this clear. Once the sperm binds to the zona pellucida, you're calling that the acrosome reaction. Okay, binding of the sperm to the zona pellucida is inducing this reaction. But when you're talking about the stages of fertilization, all of them they involve uh, penetration through the corona radiator first, the zona pellucida, and then lastly fusion of the gametes. So the sperm will now be able to penetrate these layers, and then fertilization will occur through fusion of the nuclei themselves. Okay, so those stages there they're illustrated in this picture, as we are saying. We'll have, for those who have been following, we know that the oocyte does not complete meiosis 2. Okay, it is in metaphase of prophase 2. It is arrested. This is the second arrest in metaphase of prophase. Metaphase of meiosis 2. Okay, so once it is in uh, that state, we have the coronary radiator, the zona pellucida there, those structures that we're able to see, and then the sperm come, and then once the sperm has been able to penetrate these layers, it meets the zona pellucida, you're going to have that acrosome reaction happening, and then it's able to penetrate like that, and then as you can see the sperm in there. As the one thing I forgot to mention, as the sperm is binding uh, to this zona pellucida, or rather let's just say the whole oocyte, that's when the second arrest is lifted. Okay, so this oocyte will be moved from its second arrest, and then it will be uh, the process of meiosis will continue and it will finish. Once it finishes, you will have formation of what is known as the pronucleus. That is just the nuclear the nuclear material condensing and arranging itself, getting ready to combine with the other genetic material that is coming from the sperm. So once it arranges itself into that configuration, the you're going to find the male pronucleus also being formed because the sperm has entered there, and then the centrosome as well. You see them uh, being arranged in that manner, but that's not of particular importance at this point. So at this point, basically all that is left is fusion of the male and female pronuclei. Once they fuse, you're going to find uh, uh, fertilization has occurred. Once it occurs, then you're going to have these stages here of divisions. Okay, this is the first division that you're going to have, and these divisions are meiotic and not these divisions are mitotic and not meiotic. So it was the zygote that we are forming after formation of uh, after meeting of the male and female pronuclei. It's the zygote that we form, the single cell organism known as a zygote, is now going to start dividing. And then once it starts dividing there, it's going to produce this structure that we are seeing here. Okay, so basically what I'm from explaining is basically in a nutshell fertilization. Okay, so there's also another reaction to take note of as fertilization takes place. Okay, as you are in case you are taught to give an account about fertilization. Okay, in case you are told to give, Natasha says polar bodies. Okay, polar body. So one thing you need to know about a polar body is that remember how the polar body forms. Okay, so the polar body is going to be that. Remember during meiotic division in oogenesis, you're going to have one of the oocytes is going to get most of the cytoplasm. If we can say all, it would have been possible, but let's just say most of the cytoplasm, and then one of them gets solely to the cytoplasm that is not a viable cell, but it is still alive, so to say. But it exists in a space known as the perivitelline space. That's where it exists. So this perivitelline space, you're going to have the polar body, the one of the eggs, let's call them eggs for now, one of the eggs that did not get enough cytoplasm. So as we are completing meiosis 2 here, you find that the polar body is also finishing its meiosis 2. Okay, so during this process of fertilization, once you have completion of completion of meiosis 2 here, you're going to find that there are two polar bodies because they were dividing here. Okay, as they are dividing here, as you can see the mitotic figures here, you find that the polar bodies also finish their division, and then you're going to have two of them. Okay, as you have these polar bodies, what is going to happen to them is that as the zonal reaction happens here, once the zonal reaction happens, these polar bodies are, are going to more like just, let's just say they just degenerate. Okay, because they are not viable cells, they don't do anything. So the most important thing you're supposed to note about polar bodies is just how they are formed. Okay, just know how a polar body is formed and then you'll be set. Other than that, it does not contribute to any of the following processes after they are formed. It does not do anything in the fertilization process or in the growth of the resulting embryo. Are we okay on the polar bodies? Yes. So there is another reaction known as the zonal reaction. Okay, that happens once the sperm binds to the oocyte membrane. So once the sperm binds to the oocyte membrane, it involves a change. There's a reaction that is happening that involves a change in the permeability of the zona pellucida by release of cortical granules, which contain lysosomal enzymes. This reaction prevents polyspermy. So this reaction, what it does is that it's going to make the oocyte impenetrable to other sperms. Okay, it's going to change its configuration basically. There is also something that you hear about that is known as the fast block to polyspermy, which involves uh, switching of the membrane polarity of this oocyte. I'm sure you were talking about membrane potentials in the class before this. So that membrane potential, the normal membrane potential is reversed so that it blocks sperms that are entering in a fast uh, time frame, so to say. So what you want is that once one sperm binds, another sperm is not able to do the same thing. So another sperm is not able to penetrate. So since there are millions of sperm that are released at a time, how do you do that in a millisecond? How do you do that in the quickest time possible? There is first what is known as the fast block. The fast block happens basically before the zonal reaction, and then it's going to involve reversal of the membrane polarity of this oocyte that is going to prevent further binding of other sperms. And then after that, you're going to have the zonal reaction. The zonal reaction happens after the sperm binds as well, but you can say it happens after the first block. The first block is not documented in the book that is recommended for you, so it's just something that you might find as you're writing a test or something, but just keep it at the back of your head. It's not documented in either the slides or the book that is recommended for you guys. But yes, then the zonal reaction, on the other hand, it involves release of what are known as cortical granules. The cortical granules are found just under the membrane there. Okay, and then once the sperm binds, you're going to find that they are released, and then what they do is that they also alter the properties of the membrane to enable it to resist penetration by other sperms. That's basically what it is uh, doing. Why are we doing all of this? For the first block and the zonal reaction, we're preventing polyspermy. We don't want multiple sperm fertilizing this egg because it will give us a zygote that has more than the normal number of chromosomes, and obviously that pregnancy will have to be terminated. All right. So at the end of the day, after fertilization occurs, we have a few results. Okay. Once fertilization has occurred, the first result that we have is the resumption of the second meiotic division of the oocyte. Once the sperm binds, the oocyte will continue its meiotic division from the metaphase that it was arrested in. The other thing is restoration of diploid chromosome number. We know that the two gametes are haploid, but at the end of fertilization, we know that the zygote will be diploid because uh, it will have a complete set of chromosomes from the mother and from the father's gametes. And then there's also determination of sex of the new individual. At this point, you can tell that it's a lady or it's a girl genetically. Okay, genetically you're able to tell. What did I say? Say a lady or a girl? Okay, it will be a lady or a gent, depending on, yeah. Okay, then the other thing that happens is the initiation of cleavage. What is cleavage? It is a series of the mitotic divisions that result into the increase in number of cells known as blastomeres. So once we have this single-celled organism known as the zygote, we have to divide. The cell has to divide and become a full human being at some point. So cleavage has to occur. This is a series of mitotic divisions that result into increased number of cells known as blastomeres. Okay, so the blastomeres undergo compaction, which is tight addition between the inner cell mass. So what I'm saying is basically uh, the zygote will start to divide, the cell number will start to increase. But the interesting feature about this cleavage is that you're going to find that the cell number is increasing, but the oocyte, sorry, let me not use the word oocyte anymore because it's a zygote, but the cell size is not increasing. So the reason why that occurs in that manner is because the divisions are so fast. The division is so fast, we don't have enough time. We don't have enough time for the other cells to be getting enough cytoplasm. We don't have enough time for the other cells to be getting enough cytoplasm. So once the divisions are occurring, they are happening so fast that the cells that are resulting, they are smaller cells, but they are increasing in number. That's what happens during cleavage. And then once those cells are being, uh, once those cells are increasing in number, you find that there is what is known as compaction that has to occur. They have to be compacted together, basically, and this is a difference between a compacted, uncompacted, and compacted. So I swapped the labels. B is supposed to be the compacted, and then A is supposed to be the uncompacted, as you can see there. So these are just swapped. Okay, so that's compaction. Once compaction occurs, there are certain cell stages, and these cells are called a different name depending on the number of cells, basically. So before it's 16 celled, it's called they're called blastomeres, like we said. The cells are known as blastomeres. Once we reach at 16 cell stage, we're calling it a morula. Okay, the morula will have around 16 cells. Okay, around 16 cells, and then it will have what is known as an inner cell mass and an outer cell mass. That means there are cells that are found on the outer part, and then there are cells that are found on the inner part. What will those cells form? They will form those that are in the inner part. They're the ones that are going to form the embryo, and we call those the embryo proper. Okay, they will form the embryo proper. That means they'll form the embryo at the end of the day. The outer cell mass will form the placenta, and we'll see how that will occur as we go on. Okay, so the modular stage, we have talked about it. So once that modular uh, starts to grow a little bit in size, there is what is known as blastocyst formation. Okay, so what happens during blastocyst formation? The morula will start to accumulate some fluid in the intercellular spaces. That means the spaces between the cells, the spaces between those cells of the morula, this one, they will start to fill up with some fluid. And then once that happens, that fluid filled space will be called the blast or seal, and then the morula will now be called the blastocyst. Okay, so this is what we are talking about. Initially, you will have something without the cavity, and we're calling that a morula. And then that morula has an inner cell mass and an outer cell mass. But there will be a space that will be formed, and then the fluid will start accumulating there. Once that fluid starts accumulating, you're calling that space that the fluid is accumulating in, you're calling it the blastocele. Okay, that space is called the blastocele. And then the whole structure now changes the name from a modular to a blastocyst. Are we together up to this far? Are we following up to this far? Do we know how a blastocyst forms? You can get reactions or anything in the comment section. Are we together? Do we know how the blastocyst forms? Know what a blastocele is? I'll take the silence as a yes. As people are not responding. I'll take the silence as a yes. Okay, someone raised their hand. You can go ahead, Namakau. All right. So I'll repeat. I'll repeat about how the blastocyst is formed. So I said, take you back here. So the zygote starts to divide. into a number of cells. Then compaction happens between those cells, and those cells at that point we are calling them blastomeres. Okay, we are calling them blastomeres. And then once the divisions reach at a certain point where the cells are now around 16, at the 16 cell stage, that blastocyst, sorry, let me just repeat, let me just go back a bit. So that morula, once it is at the 16 cell stage, we're calling it a morula. The blastomeres that are dividing from the zygote, once they reach 16, the number of blastomeres, once they reach 16, we're calling it a morula. So that morula will start to have some distinct cell boundaries. We'll have some cells that are on the outer cell mass and some cells that are on the inner cell mass. Okay, so those that are on the inner cell mass, we're calling them the embryo proper, and then those that they will make the embryo proper rather, and then those that are on the outer cell mass, they'll form the placenta. So that morula, the 16-celled organism, let's call it an organism for now, will start to accumulate some space, some fluid in a space, and that space will call it the blastocele. Okay, they will start to accumulate some space between the cells. Between those cells, will start to get some fluid accumulating like that. There, that space that you are seeing. So that space, we're now calling it the blastocele. So because we have this blastocele here, the name of this uh, organism is changing. I don't know what best to call it, but the whole thing will now be called a blastocyst. Okay, once we are there, the blastocyst has a distinct inner and outer cell mass, like we said before. So this that we are seeing here is the inner cell mass, and the outer cell mass is this thing we are seeing on the outside there. Okay, so that arrangement, once we have that arrangement, we'll have this thing by the time it's reaching this particular structure, it will be reaching the uterus. So implantation has to start happening there. And so this is it's more like a different pictorial diagram to show you what I'm just from saying. So you have the inner cell mass and the outer cell mass. Okay, so the inner cell mass is called the embryoblast at this point. Okay, it's called the embryoblast. And then the outer cell mass is called the trophoblast. The outer cell mass is called the trophoblast. So I hope we are together. The blastocele now has some pretty interesting names. The inner cell mass will now be called the embryoblast because that's what will form the embryo. And then the outer cell mass will be called the trophoblast. And then this is the structure that will start implanting in the uterine stroma by uh, by the end of week 1. Okay, just after around 5 to 7 days, it will start implanting. That is what is happening here. So the trophoblast cells will be the ones that will start invading the uterine stroma. Or this is just the uterine stroma, just the uterine wall. That's what we are referring to as the uterine stroma. Okay, so at that point, we are done with week 1. So that is what happens all the way up to week 1. I hope we'll be done with week 2 in 30 minutes because we are supposed to be done by 10:30. But yes, that's what is basically happening. So by the end of the week 1, this is the structure that you have, and I'm sure we have well explained how all of this is coming about. So now we go on into what is happening in week 2. Okay, so in week 2, the main thing that we have happening is formation of the bilaminar gem disc. That's what is happening in week 2. So it is also known as the week of twos because of the events that we'll see as we go on. And I'm sure some of you have seen questions like why is it called the week of two? Okay, so what is the difference between someone says Natasha? Natasha Mutambo says, what's the difference between the blastocele and the blastocyst cavity? It's the same thing. The blastocyst cavity is the same thing as the blastocele. So the blastocyst cavity, you're simply saying the cavity that is found in the blastocyst. That's the blastocele, but it has a specific name known as the blastocele. All right. So week 2, formation of bilaminar gem disc. So at week 1, at the end of the week 1, um, we are having the blastocyst implanting. That's where we leave off week 1. So at the start of week 2, we're supposed to see structures that are now changing, starting from where we left that blastocyst off. So let's see exactly what is happening in week 2. Okay, so at this point, we are saying that it has reached the uteroendometrium, so it has reached the uterus at this point. And then implantation is underway. Implantation has started in week 2. So it will be called the week of twos, and the main thing that happens is formation of what is known as a bilaminar disc. Okay, so implantation, the other name for it is called nidation. However, you might choose to pronounce it nidation, just get the spelling correctly. Yes, so this is where we left it off in week 1. Before implantation starts, one thing you need to know is that there has to be a process known as hatching that has to occur. Hatching, similarly to what happens to chickens, the embryo has to hatch. But what is this hatching in terms of embryology, human embryology? Hatching is disintegration of that zona pellucida that we are talking about. Remember that the zona pellucida was still there all this while, but now it has to disintegrate. And how does it disintegrate? We didn't mention. So this is how it happens. So once it reaches there are chemical signals that allow it to disintegrate, and we're calling that process of disintegration of zona pellucida, we're calling it hatching. Why does it occur this way? So hatching has to occur specifically when the embryo reaches the uterus to prevent uh, the blastocyst implanting elsewhere apart from the uterus. So once it reaches the uterus, there will be chemical signals that will allow it to hatch and then it will implant there. If not, then you might find it implanting in the wrong place, like the fallopian tubes, and you're calling that an ectopic pregnancy. That is uh, why it's important for hatching to take place, where it takes place, and how it takes place. So day 8. So we're done with week 1, and now we're day 8. That is, we're entering the second week. The blastocyst at this point has started implanting itself into the uteroendometrium, and it is in the uh, partially embedded in the endometrial stroma. Okay, so at this point, sorry, let me just gather my thoughts where I was. Yes, so it's partially embedded in the endometrium, and you can see a few things occurring at this point. You see, okay, sorry, my network scattered, received the call. So at this point, what we were calling the trophoblast would change and develop into two structures. Okay, the trophoblast will develop into two structures, and also the embryoblast will develop into two structures. It will divide into two structures. Let's call it that for now. So let me just take you back a little bit. We have the embryoblast here, don't forget, and then the trophoblast here. The outer cell mass being the trophoblast, and the inner cell mass being the embryoblast. And we are saying that the embryoblast splits into two structures, and then the outer mass cell mass, which is the trophoblast, also changes into, or rather, it also splits into two structures. Which are those structures? The trophoblast will change into the syncytial trophoblast, and then the the cytotrophoblast as well. So the trophoblast, the outer cell mass, divides into the syncytial trophoblast and the cytotrophoblast. Where are these structures? Here we are seeing them. So this is the syncytial trophoblast, the one that is in green, like the darker shade of green, and then the cytotrophoblast is the one that is there in the lighter shade of green. So why is this one called the syncytial trophoblast, and what is it exactly? So the syncytial trophoblast is the is a it's more like an undifferentiated group of cells. You won't find distinct cell boundaries in the syncytial trophoblast. It's more like a mass of cells that don't have distinct cell boundaries between them, and then it interfaces directly with the endometrial stroma. That is why you are calling it a syncytial trophoblast. It forms a syncytium with the endometrium. So it's like an interface between the embryo and the endometrium of the mother. That's how we are going to find structures that enable exchange of materials between the mother and the developing baby. That's why it's called the syncytial trophoblast, because it forms a syncytium. All right. Then the cytotrophoblast is the one that is lining on the embryo side there. Okay, so those are the true structures that the outer cell mass splits into. But the inner cell mass will split also into two parts, known as the epiblast and the hypoblast. Okay, the inner cell mass, the ones that is color coded blue, will split into the epiblast and the hypoblast. The hypoblast is the one that we are seeing down there, color coded in yellow, and then the epiblast is the one color coded in blue. Okay, so that's how we are changing from this structure to this structure here. Okay, and then another cavity will appear in the epiblast. Okay, so remember, first we have this cavity here, and we know where this cavity is coming from. It's coming from when it was still a blastocyst. This one, it's this cavity, and still here it's called the blastocyst cavity, which is the blastocyst, the same thing. But another cavity will appear in the epiblast. Okay, one of the layers that the embryoblast splits into, the epiblast, and then that cavity will be called the amniotic cavity. That's where the amnion comes from. Okay, so the cells lining the amniotic cavity and adjacent to the cytotrophoblast are called amnioblasts. So what do we mean adjacent to the cytotrophoblast? That means these cells that are next to the cytotrophoblast. The cytotrophoblast is the one that is in light green, and then the ones that are lining the amniotic cavity there, we're calling them amnioblasts because they are related to the amniotic cavity, of course. Okay, so that is basically what is happening there uh, on day 8. Okay, so where it implants, usually in between the uterine glands, that's where it usually finds a place to implant in the endometrium. Okay, we have 12 minutes to go. So we move on to day 9, from day 8 to day 9. What is happening on day 9? We have this thing being uh, more deeply embedded in the endometrium. We have a few extra things happening. Number one, let's go back to what is known as the syncytial trophoblast. What we say the syncytial trophoblast. The syncytial trophoblast starts to uh, have some spaces appear in it. Okay, these spaces will be called lacuna or lacunae, however you might choose to pronounce it, but just don't forget the pronunciation, or rather the spelling. Okay, so these lacuna will develop uh, in the side syncytial trophoblast, the outer cell mass, or the syncytium. And then another thing that will happen again is development of what is known as the Husser's membrane, or the exocoelomic or exocoelomic membrane lining the blastocyst cavity. So remember the blastocyst cavity, where we are taking it from. we're going to find that that cavity will start to be lined by a membrane, this thing color coded in yellow. And this membrane is being called the Husser's membrane or the exocoelomic membrane. Okay, so this exocoelomic membrane, some will say, depending on which literature you read, some will say that it's produced by the cells of the hypoblast and the like. Some will say that the cells themselves, the hypoblasts, will migrate. Some of them will migrate, will line this this exocoelomic cavity, and they will produce the extra membrane that is being called that. But I don't think that is of particular importance. Just know what it is. In this case, we're calling it the Husser's or the exocoelomic membrane. And once that happens, the blastocyst cavity will now change from that name. We'll call it either the exocoelomic uh, uh, cavity. We'll call it the extra. Okay, let me not mix up those terms. We're going to call it either the exocoelomic cavity or the primitive or primary yolk sac. So there are three names that you can call this thing after development of the Husser's membrane. You call it the exocoelomic cavity, the primitive or primary yolk sac. That's where it comes from. So the names, I know the names are becoming plentiful, but just bear with anatomy for now. They will make sense. Just keep going over them. So exocoelomic cavity, because it's lined by the Husser's membrane. Because the blastocyst cavity has developed Husser's membrane, we now call that cavity the exocoelomic cavity. Okay, I hope we're okay. After that, we move on to, because nothing much happens on day 10, because it's just mainly development of these features. They just develop a little bit further, and then that leads us to day 11 and day 12, where we have like major changes that are happening. So the major changes that you're going to see here happening is, remember those trophoblastic lacuna that we talked about, lacuna that develop in the syncytial trophoblast. They are going to start to fill up with blood. Okay, they are going to start to fill up with blood. Where is this blood going to come from? Remember that the endometrium has what are known as the sinus arteries. They're usually sinusoidal arteries. That means these arteries have some fenestrations with them. Okay, that means they have some spaces that allow blood to leak out. So once this syncytial trophoblast reaches uh, that endothelium of those blood vessels, it's going to cause the blood in those blood vessels to start tripping into the trophoblastic lacuna, into those lacuna spaces. That's what is happening on day 11 and day 12. Okay, at this point, there is what is known as a surface defect. Okay, so at day, one thing I forgot to mention, in on day 9, this thing is more deeply embedded and it's almost completely embedded, just that there is a fibrin coagulum where the thing started to embed itself into the endometrium. So when it started to invade the maternal endometrium, it starts to cover itself with the fibrin coagulum. Just normal wound healing, because at this point it's more like, you know a lesion, so to say, on the maternal endometrium. So there's going to be fibrin trying to cover up where it invaded. So at this point, on the 11 and 12, it's almost healed. And then another thing that is happening is that the spiral arteries, the ones we're calling the sinusoids, spiral arteries, they will be congested and dilated. They're the ones that are going uh, to make blood start entering the trophoblastic lacuna. Okay, well, how do they do that? The syncytial cells will erode the arteries of the endometrium and blood flows into the lacuna. So the syncytial cells, these cells here, will start to erode away some of the cells in the uh, spiral arteries that are found in the endometrium. If you check uh, a diagram on uterine cycles, you see those arteries that are found there. Those are what we're calling the spiral arteries. So those spiral arteries will be eroded away. The endometrium, the it's not the endometrium, it's the what's this? The endothelium. Yes, the endothelium. That's the cells that line the arteries. The arteries, remember, they're one cell thick and they're called an endothelium. So it is eroded away. Some of those cells are removed, in short, and then since it's one cell thick, the blood starts to come out, and then the blood starts entering the lacuna spaces. So once that happens, another thing that will happen on day 11 and day 12 is that you'll find formation of the mesoderm between the Hughes membrane and the cytotrophoblast. Remember, this is the Hughes membrane, and then this is the cytotrophoblast, the one that is color coded in light green. So in between here, that's where you're going to find the formation of the mesoderm. Okay, and that mesoderm is called extra embryonic mesoderm. Why is it called extra embryonic mesoderm? Because it is not part of the embryo. The extra embryonic mesoderm, it won't be part of the embryo because the embryo is here. Okay, the embryo proper, remember, it's here, these two layers, the hypoblast and the epiblast. So anything here is not going to be part of the embryo. Anything that is not in blue and yellow will not be part of the embryo. If anything, only the epiblast will be the embryo. The rest of these will form other structures that, of course, are necessary but will not be part of the final human being. So, in between the Hughes membrane on the inside here and the cytotrophoblast, you're going to find a mesoderm being formed. Another another type of tissue. Okay, so this tissue is now being called the mesoderm. So the mesoderm will be called extra embryonic mesoderm. The reason why we have to be specific is because uh, once we reach in week 3, you find that there are other types of mesoderms that will be present. Okay, so at that point, that mesoderm starts to have spaces in between uh, in between it as well. Okay, so those spaces in between the extraembryonic mesoderm will be called the extraembryonic cavity. Okay, so please don't mix these up. Extraembryonic cavity is found in the extraembryonic mesoderm. It develops in the extraembryonic mesoderm. Are we together so far? Yes, so extraembryonic mesoderm, some spaces start to appear in there, and those are called extraembryonic cavities. That extraembryonic cavity has now splitted this mesoderm into two again. We'll have the one on the inner part and the one that is more closer to the cytotrophoblast here. So those will now carry different names as well. So we'll have the extraembryonic splanchnic mesoderm. We'll have the extraembryonic splanchnic mesoderm. When is this meeting ended? All right, so we are back, and unfortunately, like I said, that link was only for an hour. I was supposed to finish in an hour, but yeah. So let's just do this for the next 10 minutes, and then we'll be done. All right. So where was we? All right. So we were talking about the extra embryonic mesoderm. It's splitting into two because of development of an extraembryonic cavity in that extraembryonic mesoderm. So the extraembryonic mesoderm will now have extraembryonic somatic mesoderm, which is closer to the cytotrophoblast, and then the ones that is on the inner part, the one that is on the inner part will be called the extra embryonic splanchnic mesoderm. Okay, so I hope I didn't leave anyone behind there. I just realized I was talking without the mic being on. So yeah. All right. So let me just repeat what I said. So someone has the mic on. So if you joined the mic on, kindly mute it. All right. So I was saying that there is formation of the mesoderm between the uterus membrane and the cytotrophoblast, and that mesoderm is called the extraembryonic mesoderm. This mesoderm develops. Natasha, kindly mute your mic. Okay, so the extraembryonic mesoderm will develop a cavity, and then this cavity will split uh, the extraembryonic mesoderm into two. So that cavity is known as the extraembryonic cavity, of course, because it is in the extraembryonic mesoderm, and then that mesoderm will split into two. The splanchnic mesoderm, which is on the inner part here, while the one that is closer, or rather the one that is continuous with the cytotrophoblast, is called the somatic mesoderm, the extraembryonic somatic mesoderm. All right, so uh, this extraembryonic mesoderm, this is basically explaining what I'm just from saying, the two layers that now exist in the mesoderm. It's just from what I'm from saying. So day 13, what happens on day 13 is what you basically see here is enlargement of this extraembryonic cavity that we are seeing here, that is developing, that is splitting the extraembryonic mesoderm. It's being enlarged like this. While what we said was the yolk sac in the first place, the primitive yolk sac, remember from this picture, the primitive yolk sac or the primary yolk sac, that was still the primitive yolk sac here, or the exocoelomic cavity, is being pinched off, it's being reduced in size. As it is reducing in size, you find that now the extraembryonic cavity increases in size. So as it is being pinched off, its remnants are what is known as the exocoelomic sized. It's just it's more like a piece of tissue that remains uh, as it is being removed from the primary yolk sac. So as the primary yolk sac is being reduced in size, one of its uh, remnants, so to say, as it is regressing, it remains as the exocoelomic size. Okay, so here you find that there is um, there's some name changes again that is happening just because of this change that we talked about. So what are the name changes and the other things that will be happening? So the primary cavity or the primary yolk sac after this regression is now known as the secondary or definitive yolk sac. So the primary yolk sac will now be called the secondary yolk sac because it has because of these changes that we're just from talking about, the enlargement of the extraembryonic cavity, this one, or the chorionic cavity, that's the other name. So don't be, in case you find the chorionic cavity, uh, don't be lost. So what we are calling the somatic mesoderm, the extraembryonic somatic mesoderm, the one that is continuous with the cytotrophoblast, is now also called the chorionic plate. Chorionic plate. And they might use chorionic plate. One thing that your lecturer likes is to use other names for other structures just to sort of confuse you. So please learn all the names necessary that are shown on these structures that you are seeing here. So primary yolk sac becomes secondary yolk sac because it regresses, and as it is regressing, some of the portions remain as the exocoelomic cyst. And then the extraembryonic or the somatic mesoderm, or the extraembryonic somatic mesoderm, is also called the chorionic plate, another name. And then what other change are we seeing here? What other change? What other change? Okay, so there's development of what is known as the amniotic, sorry, not the amniotic cavity, but the connecting stalk. We have the connecting stalk here. So, how does the connecting stalk form? Remember that the mesoderm forms between the embryo proper here in this case and the cytotrophoblast. So, as it forms there, the space is the space of the extraembryonic cavity is going to enlarge all the way here, but it will just stop at a certain point there, and that point will remain as the connecting stalk. So they'll ask you a question: at which point is the somatic and splanchnic extraembryonic mesoderm? Where do they meet? Where do they meet? Okay, so if you follow the splanchnic mesoderm, sorry, this is the somatic. So if you follow the somatic mesoderm, you find that it will meet at the connecting stalk there. If you follow the splanchnic as well, you'll meet there at the connecting stalk. So this is the point at the connecting stalk where the splanchnic and the somatic mesoderm meet. Another name they might call it is the somatopleuric and the splanchnopleuric membrane. Sorry, the somatopleuric and splanchnopleuric mesoderm is what we are calling these two mesoderms. And where do they meet? At the connecting stalk there. This connecting stalk, we'll see, ladies and gents, that it will become eventually the, who can guess what it will become? It will become the umbilical cord, of course. All right, another thing that changes, I think this is the last thing to change, it will be the villi. Okay, primary villi will start forming. So in the cytotrophoblast, we'll see columns of cells start to form. These are what we are calling primary villi. These villi, we'll see, they are very important in week 3 because in the mesoderm, you find that that's where some blood vessels will start to develop. These blood vessels will have to interface with this blood that is here because at this point, the embryo is still small, yes, and then diffusion is enough to take the nutrients from this blood that is here all the way to the cells. But imagine this thing gets big and then the blood is not able to deliver the nutrients properly all the way up to the embryoblast here. You can see that it's being isolated now. So because of that, these primary villi will be very important because that's where the blood vessels will pass. There will be a good network of blood vessels that will take the blood all the way to the embryo proper there, as we'll see in week 3. So primary villi, we are seeing these develop on day 13. Okay, so that's that. Day 13, another day 14. So this is supposed to be day 13 and 14. Things that happen within that time period, within those days. So the clinical applications that you might want to know about this is ectopic pregnancies, which already talked about when we are dealing with week 1, improper implantation or implantation at the wrong site. So maybe something that I might want you to go figure out is where else the embryo might implant except for the uterus and the fallopian tube. There's also other places that it might implant. So go check those out. Okay, so go check those places out. Yes, so I was saying go check out those places where the embryo can implant elsewhere the apart from the fallopian tubes and the uterus. And then one other thing that you need to note as well is that remember that there is that structure known as the corpus luteum. Remember we said that the corpus luteum produces progesterone, and progesterone is important for maintaining the thickness, or rather for maintaining the uterine lining as it is. So because the uterine lining has to be maintained uh, in its thickness the way it is, it has to be maintained so that it keeps providing the optimum environment for that embryo to be growing. So the corpus luteum produces progesterone that helps for it not to be degraded. So because let's say the first scenario, pregnancy does not occur, of course the corpus luteum will degrade. But the production of HCG by the syncytial trophoblast prevents that from happening if pregnancy occurs. So when pregnancy occurs, the syncytial trophoblast also produces HCG hormone. This syncytial trophoblast that we were talking about, it produces HCG hormone, also known as human chorionic gonadotrophin hormone, that keeps uh, the corpus luteum from degradation, and then the progesterone can keep being produced. That means the uterine lining will remain the way it is, because if it were to be removed, that means menses will happen, and then obviously the pregnancy won't hold. But that does not happen, it does not degenerate, and then that is why you find that the pregnancy is maintained. But then you find that at this point, as we're entering week 2, because this is day 13, day 14, 2 weeks have passed. 2 weeks have passed from day 14 when we started this journey. We find that that is day 28. At this point, you find that some ladies who are pregnant at this point, you find that they still get to experience some blood, you know, some blood being coming out at that particular time. So what is the explanation between that for that rather? So you find that the blood that is in these trophoblastic lacuna, as it is entering there, some of it can seep out and it may appear as light spotting and such a thing. So some ladies might not realize that they're pregnant at this point until it's like an entire month later, or until they miss their second period, because at this one they might think that they have attended that particular month, but it was just light spotting and the like. And usually, uh, if for example it's a common occurrence for this lady to have light menses, she might just dismiss that. So most people will find that they figure out that they are pregnant after they miss the second. Uh, they miss the second. Yes. All right. So that will be it for today. That will be it for today. Unless there is unless there is any question.